Hello and welcome to Sam Biology class. Friends, today we shall discuss about the principle and applications of adsorption chromatography. Before that, I request all of my friends, please subscribe my YouTube channel. Adsorption chromatography is based on the principle that certain solid materials, collectively known as adsorbents, have the ability to hold molecules at their surface. This process is known as adsorption. It involves weak and non-ionic attractive forces of van der Waals and of hydrogen bonding type. Adsorption occurs at specific sites called adsorption sites, which remain occupied by the molecules of the element or of the analytes in a proportion determined by the relative strength of interaction. During separation, as the element constantly flows through the column, differences in this binding strength eventually leads to the separation of different molecules. Two differing factors are exploited for the separation of molecules by adsorption chromatography. One is the different degree of adsorption of various molecules on the surface of adsorbent, and the other is their varying solubility in the solvent or the mobile phase. Stationary phase. In this technique, the stationary phase is a suitable adsorbent. Silica is a typical adsorbent. It has silanol groups on its surface. These groups are weakly acidic and therefore can interact with polar functional groups of the analyte or of the solvent. Other commonly used adsorbents are alumina and carbon. Mobile phase. In adsorption chromatography, the mobile phase, also known as the eluent, is a suitable solvent or a mixture of solvents. In general, a solvent whose polarity is comparable to that of the most polar analyte in the sample should be used as the mobile phase. Thus, alcohols are used as the mobile phase for the separation of analytes having hydroxyl groups. For separation of analytes having carbonyl groups, acetone or esters are used as the mobile phase. For separation of analytes which are predominantly nonpolar, hydrocarbons such as hexane, heptane or toluene are used as the mobile phase. In case of gradient elution, a mixture of solvents is preferred. By applying the principle of adsorption, analytes can be separated by column, thin layer or paper chromatographic technique. This figure illustrates the principle of adsorption chromatography in column mode. It is an adsorbent which has whole molecules at its surface. Adsorption is a surface phenomenon. In this technique, first a column is packed with a suitable adsorbent. After packing, the sample is applied at the top of the column. In this example, the sample has two components, X and Y. Out of these two components, only X is selectively adsorbed on the surface of the adsorbent, hence it will be retarded. The other component Y is not adsorbed. Because of this, when the mobile phase is passed through the column, the component Y moves down quickly than X and hence we leave the column first. After this, the other component Y, which is adsorbed on the surface, can be eluted out of the column. So in this technique, based on the different degrees of adsorption, various analytes present in the sample can be separated. Hydroxylapatite chromatography is a form of adsorption chromatography. In this technique, crystalline hydroxylapatite is used as the adsorbent for the separation of proteins or nucleic acids. One of the most important applications is the separation of single-stranded DNA from double-stranded form. At low phosphate buffer concentration, both forms of DNA bind the hydroxylapatite matrix. But when the buffer concentration is increased, single-stranded DNA are dissolved and hence are eluted out of the column. But as the buffer concentration is increased further, double-stranded DNAs are released. This behavior is exploited in the technique of cut analysis. The affinity of double-stranded DNA for the hydroxylapatite is so high that it can be selectively removed from a sample extract containing RNA and protein. Another form of adsorption chromatography is hydrophobic interaction chromatography that exploit the surface hydrophobicity. This technique is useful for the purification of proteins. Normally in proteins, the hydrophilic amino acid residues are scattered on the protein surface in a way that give the proteins their characteristic properties. In an aqueous solution, these hydrophilic residues are covered by an ordered layer of water molecules that effectively mask the hydrophobic residues that remain in the interior of the folded molecules. These hydrophobic groups can, however, be exposed by the addition of salt ions, which preferentially take up these ordered water molecules. The exposed hydrophobic groups attract each other by weak van der Waals forces that result in protein-protein aggregation. In this technique, the presence of hydrophobic groups such as butyl, octyl and phenyl on the matrix result in protein matrix aggregation rather than protein-protein aggregation. Since hydrophobic interaction chromatography requires the presence of salting out compounds that facilitate the exposure of hydrophobic groups on proteins, this technique is used immediately after protein fractionation by ammonium sulfate. Another related technique is immobilized artificial membrane chromatography that resembles the hydrophobic interaction chromatography. It uses a phosphatidylcholine-based stationary phase. This technique is widely used in pharmaceutical industries to study the ability of candidate drugs to be absorbed and distributed. Applications: adsorption chromatography has been extensively used for biochemical separation for the purification of amino acids, proteins, nucleic acids, mono and disaccharides, neutral lipids, cholesterol, esters, carotenoids and phospholipids. So friends, this is all about the principle and applications of adsorption chromatography. For more updates, please subscribe my YouTube channel Sam Biology Class.